there we go. I am Michael Stubna. I am a graduate student in the Bartel Lab.
Stubna works with C. elegans, tiny translucent worms that live in soil. They can help researchers, including Stubna, answer questions about how gene expression is regulated.
Right now, I only have a couple individual animals on here, but it's possible to grow many 10 of 15 centimeter plates at a time to get tens or even hundreds of thousands of worms at a time.
While Stubna stores his worms at 68 degrees Fahrenheit, living worms can be frozen indefinitely in liquid nitrogen at negative 320 degrees.
Yeah, so manipulating C. elegans is very easy. You can pick individual worms up with this little worm pick and move them to different Petri plates. And that is done by making this worm pick, which is basically a piece of wire stuck inside of a glass tube, and you sterilize that in a flame. It's a skill that has to be learned, but it's learned very quickly, and it's one of the most basic skills in handling C. elegans.
The worms live their lives on Petri plates, which are coated with a bacteria culture. This bacterial lawn is the worm's source of food. As a C. elegans travels across the plate, it carves out a path in the bacteria culture, like a skier making tracks in fresh snow. Why raise C. elegans? These worms are helping researchers like Stubna answer questions about how levels of proteins are regulated in cells.
One mechanism that the cell has to try to activate or turn off the different sets of genes that's required for that cellular specialization actually involves microRNA.
MicroRNAs help degrade messenger RNAs, so that less protein is made from the messenger RNA. Stubna studies how particular microRNAs degrade and what causes them to degrade faster or slower.
Over the past 10-20 years, people have learned quite a few things about production of microRNAs in the cell, but a kind of bigger question mark is what then triggers the degradation of microRNAs? Why are some much more stable than others in the cell? Why do some turn over with a half-life of 2 hours and others turn over with a half-life of 3 days? So those types of questions are ones that I'm thinking a lot about in the context of the worm.